Now that we've gotten a chance to discuss the specific larger structures within a cell, such as organelles within eukaryotic cells or cytoplasm found in all cells, let's continue our journey of looking within a single cell and explore how different components of the cell are truly interconnected to allow the cell to survive and thrive. Specifically, we will be exploring how proteins, aka the doers of the cell, are made from transcribing DNA into mRNA and then translating that mRNA into functional proteins. And then those proteins can carry out their functions throughout the cell. These processes are what allow life to persist, and they are constantly taking place within your cells too, ensuring that your body continues to function so you are able to complete this lesson. So thank you, cells. And with that, let's dive in. At this point, we know the basic structure of DNA and we know where to find it. Let's now dive into its function and how understanding this can help us better understand the connectedness of different components within a cell. By this point, I'm sure you have heard something along the lines of that DNA is what makes you, you, right? It's the reason your eyes are blue, or your hair is black, or you're somewhat tall. While DNA does contribute to these features, how does a line of code made up of A's, T's, C's, and G's cause this? It's actually a bit more complicated than most let on. Actually, DNA is just cellular instructions on how to make certain proteins. The proteins are what that contribute to your phenotype. Phenotype being your observable characteristics, right? Such as your eye color. So when people talk about gene expression or expressing DNA, right, what I really mean is that a gene within a DNA sequence was successfully used to make a functional protein. However, the DNA alone cannot express itself. DNA cannot do anything, right? It's just a set of coded directions. So the whole process of making a protein from the information from the DNA sequence is orchestrated by different proteins that participate in different parts of the process of gene expression or of expressing a gene. I like to think of this kind of like baking a cake in a bakery, right? To bake a cake, you have your cookbook, a specific recipe within that book, a baker, and your ingredients for baking. In this analogy, the whole cookbook represents a full strand of DNA, while a specific recipe within that book for a specific cake represents a gene. Genes are a length of DNA that code for a specific protein. The baker represents the ribosomes, which read the directions to make proteins. Now, just as a baker would want to make sure that their special cake recipe is preserved for many years to come, the cell also works to ensure that their DNA instructions do not degrade over time. This is where transcription comes in. Our baker friend takes the recipe from their cookbook and makes a photocopy of the recipe so they can use it in their messy kitchen without worrying. Similarly, for the directions of DNA to leave the nucleus, right, as though they were leaving the cookbook and into that messy kitchen, for the DNA directions to leave the nucleus, it is first copied or transcribed onto mRNA. Tying this back to Unit 0 material, do you remember any other reasons why DNA is transcribed into mRNA? So you can think of RNA polymerase in this analogy as the copying machine, right? And you'll see why in just a second. And then the ribosome reads the mRNA one codon at a time, as a baker would read the recipe one step at a time. Each codon corresponds to an amino acid, just like each step in a recipe refers to a specific ingredient. All right? Keeping in mind that the specific ingredient listed in the directions is not the ingredient itself, right? So in our directions, if it says get sugar, There isn't sugar physically in the cookbook. We just know that we should now go get the ingredient sugar because of what we read. Same thing in how codons correspond to amino acids. The amino acids are brought to the ribosome by tRNA, or transfer RNA. So you can think of these tRNA molecules like little interns helping out the baker. They deliver the ingredients to the baker, which in this case is delivering the amino acids to the ribosome. The ribosome then joins the amino acids together to make a polypeptide, just like a baker would mix ingredients together to bake a cake. So there's your analogy. Let's now go into a bit more detail on how this works within cells. So, for your edification, this sequence that we are discussing today is referred to as the central dogma. The central dogma is an explanation of the flow of genetic information within a biological system, specifically dictating that DNA is transcribed into RNA, which is then translated to make proteins. So let's say we are looking inside of a T cell within a eukaryotic organism, all right? A T cell is a type of white blood cell that contributes to your immune system. So let's say this T cell needs to produce cytokine proteins, which are proteins that help regulate the activity of the immune system. How will the cell do this? Well, it will all begin in the nucleus of that eukaryotic cell. Here we have our directions in the form of DNA. DNA is too large to leave the nucleus, and the cellular machinery used for reading genetic code and using said code to make proteins, aka ribosomes, are out in the cytoplasm of the cell, right? They're outside of the nucleus. Therefore, DNA must be transcribed into RNA so the directions to make cytokine proteins can reach the ribosomes outside of the nucleus. Remembering back to Unit 0 material, RNA sequences are much shorter than DNA, and if we are transcribing just a small part of the DNA onto mRNA, then of course that will be a much shorter bit of coded information to transfer. So that will be small enough to leave that small nuclear pore in the nuclear membrane. This transcription process, transcribing DNA into RNA, takes place in 3 steps: initiation, elongation, and termination. All genes, including the gene that codes for the cytokine proteins, have something called a promoter. As the name would imply, promoters help promote transcription by essentially marking where a gene begins and where proteins should bind to the DNA to initiate transcription. This is the first part of initiation. In eukaryotes, promoters are a sequence of DNA that include a sequence called a TATA box, which reads TATAAA, right? Thymines and adenines, along the coding strand of DNA, and is easily recognized and bound to by transcription factors. This promoter/TATA box is found just upstream of the gene in the DNA sequence that we want to transcribe. Initiation is when an enzyme called RNA polymerase binds to the promoter. This signals the DNA to unwind, so the RNA polymerase can read the bases in one of the DNA strands. Now, I know you just heard the word enzyme, so immediately some of you might feel like we're facing a sort of chicken or egg situation here. Yes, we need proteins, such as RNA polymerase enzyme, to make proteins. So how did we get the original proteins that help us make more proteins? Well, as it turns out, these essential proteins that help with gene expression are actually gifted to us by our biological mothers. So in addition to their DNA, a biological mother's egg contains these essential proteins for gene expression. Thank you, moms. Okay, so RNA polymerase has bound to the DNA, and the DNA has unwound. The enzyme is now ready to make a strand of mRNA with a sequence of bases that are complementary to the original DNA strand, which brings us to step 2 of transcription, elongation. Elongation is the elongating or addition of nucleotides to the RNA strand. RNA polymerase reads the unwound DNA strand and builds an RNA molecule using complementary base pairs, where C's pair with G's and A's pair with U's. As it turns out, RNA does not have thymine. Instead, it has something called uracil. So during the elongation process, an adenine in the DNA is paired with a uracil, or a U, in the RNA. Now, RNA polymerase only works on one strand of DNA. So to help keep the two strands of DNA straight, we refer to one strand as the sense strand and the other strand as the antisense strand. So when you hear those terms, this is all they're referring to, right? The strand that is transcribed by RNA polymerase is called the antisense strand, and it is complementary to the RNA sequence that's being built. The strand that is not transcribed is called the sense strand. If RNA polymerase carries out its job correctly, the sense strand should be basically identical to the RNA sequence that's being made, except there are U's in the places of where T's were in the DNA sequence. Make sense? So once we have our RNA strand, the next step is step 3, termination. Termination is the ending of transcription, and it occurs when RNA polymerase crosses a stop or quote-unquote termination sequence in the gene. The RNA strand is complete and it detaches from the DNA. DNA has been successfully transcribed into RNA, meaning the directions are copied and almost ready to leave the nucleus. Yay! So, circling back to our original scenario, within the nucleus of our T cell, the gene for a cytokine protein was read and transcribed into RNA by RNA polymerase. Now we have a copy of the directions for making those cytokine proteins that are small enough to exit the nucleus through the very small nuclear pores. But before the RNA can leave, it must undergo some modifications, because once it leaves the nucleus, it's going straight to the ribosome to be translated into proteins, and we need to make sure it's ready for the journey. So, at this point, we refer to the RNA sequence as quote-unquote pre-mRNA, but once it's ready to leave the nucleus and be translated, we will refer to it as messenger RNA, or just normal mRNA. The first modification that takes place is a 5' cap is added to the 5' end of the pre-mRNA strand. While DNA is being transcribed into RNA, a 7-methylguanosine cap is added to the 5' end of the growing RNA transcript by a phosphate linkage. This addition of this phosphate group is important because it protects the newly forming RNA from degradation. It also helps with protein synthesis, or more specifically, translation, because factors that are involved in translation recognize this cap and help initiate translation by the ribosome. The next modification that must be made is the addition of a poly-A tail to the 3' end of the strand. After the elongation step of transcription, an enzyme called poly-A polymerase adds a string of approximately 200 adenines to the 3' end of the RNA transcript. We call this series of 200 adenines the poly-A tail. And the poly-A tail further protects the pre-mRNA from degradation, and it is also the binding site for a protein necessary for exporting the processed mRNA out of the nucleus and into the cytoplasm. Now, something wonky about eukaryotes is that their genetic code actually contains a lot of quote-unquote extra information. So to make sure only the important bits leave the nucleus and are translated into proteins, something called RNA splicing must also occur before the mRNA strand can leave the nucleus. Eukaryotic genes are made up of exons, which correspond to protein coding sequences, and intervening sequences called introns. The X in the word exon signifies that they are the part of the gene that gets expressed, while the int in introns denotes their intervening role in the sequence. Intron sequences in mRNA do not encode functional proteins. Introns must be removed to make a translatable mRNA. The process of removing introns and then reconnecting the exons is called splicing. The splicing process is catalyzed by protein complexes shown on the screen in purple called spliceosomes. Spliceosomes are composed of proteins, which are easily seen on the screen here, and RNA molecules that are too small to be seen here, but are associated with these proteins, small nuclear RNAs, or snRNAs, because they are small bits of RNA that reside in the nucleus, so small nuclear RNA. The beginning and end of each intron is marked with specific nucleotides. GU at the 5' end and AG at the 3' end of the intron. Spliceosomes recognize these sequences at the 5' and 3' ends of the intron, so they know where to make cuts to remove the intron from the sequence. So, to reiterate, transcription occurs within the nucleus. After DNA is transcribed into RNA, before the RNA can leave the nucleus, the RNA must first 1. Have a 5' cap added to the 5' end. 2. Have a poly A tail added to the 3' end. And 3. All of the introns must be removed from the RNA sequence through a process called splicing. After all of this is complete, we now have an mRNA strand that is ready to leave the nucleus and be translated into a protein. Translation involves decoding a messenger RNA, mRNA, and using its information to build a polypeptide or chain of amino acids. For most purposes, a polypeptide is basically just a protein, with the technical difference being that some larger proteins are made up of several polypeptide chains. So similar to transcription, the process of translation also takes place in 3 key steps: initiation, elongation, and termination. In contrast to transcription, though, the process of translating mRNA into proteins takes place within ribosomes, which, if you remember from our previous discussion, ribosomes can either be found free-floating in the cytoplasm or attached to the rough endoplasmic reticulum. Now, to understand these 3 steps of translation, initiation, elongation, and termination, we must first break down the definition of a new term, codons. Codons are basically nucleotide triplets. That is, they are a sequence of 3 nucleotides, which together form a unit of genetic code in a DNA or RNA molecule. Ribosomes read mRNA 1 codon at a time, and each codon codes for 1 amino acid. So, the order of the codons in an mRNA sequence determines the order of amino acids in a polypeptide chain. This is where the decoding happens and why this process is called translating, because it's translating the genetic code in codons into amino acids, which are then bound together one at a time to form that polypeptide. So, to ensure that the whole sequence of mRNA is translated into a protein, meaning we are making sure that translation is not somehow starting in the middle of the mRNA sequence, the ribosome reads for a quote-unquote start codon. A start codon is the sequence AUG, found at right after the 5' cap, at the beginning of all mRNAs that are ready for translation. Initiation is when we have a ribosome divided into small and large subunits, an mRNA sequence, and an initiator tRNA that is carrying the amino acid methionine, which corresponds with that AUG codon. When we have all of these components coming together to form that initiation complex, that is when we have initiation of this translation process. So the initiation complex is the molecular setup needed to start making a new protein made up of all the units I just described. So to break this down, first the tRNA carrying methionine attaches to the small ribosomal subunit. This tRNA also has an anticodon, UAC, associated with it, which just so happens to be complementary to AUG, right? U associates with A is complementary to U, and C is complementary to G, which is our start codon. So this is how initiation takes place in translation. Together, the ribosome binds to the 5' end of the mRNA by recognizing the 5' cap. Then it quote-unquote walks along the mRNA in the 3' direction, stopping when it reaches the start AUG codon. The tRNA's UAC anticodon then binds to the AUG start codon, and the second step of translation is ready to take place: elongation. Within the ribosomes are 3 tRNA binding sites, labeled A, P, and E. During the elongation step of translation, tRNAs enter the acceptor site, or the A site, and the anticodon on the base of the tRNA pairs with the codon in the mRNA, complementary pairing. A peptide bond then forms between the amino acid from the A site with the amino acid from the P site. The P in P site technically stands for peptidyl tRNA binding site, but I like to think of it as the peptide bond site because this is where the tRNA goes after its amino acid forms a peptide bond with the previous amino acid. The formation of each peptide bond is catalyzed by an enzyme called peptidyl transferase. After the amino acid in the A site forms that peptide bond with the growing polypeptide chain, meaning with the amino acid adjacent to it, the ribosome will move 3 nucleotides, so 1 codon, towards the 3' end of the mRNA. This movement transfers the tRNA with the growing polypeptide to the P site and allows the quote-unquote empty tRNA to then exit at the E, or exit site, of the ribosome. During translation elongation, the mRNA template provides tRNA binding specificity. As the ribosome moves along the mRNA, each mRNA codon comes into register, and specific binding with the corresponding tRNA anticodon that carries a specific amino acid is ensured. Let's watch this elongation process of translation in action. Termination of translation begins when a stop codon is in the A site of the ribosome. Stop codons include the sequences UAA, UAG, or UGA. These 3 sequences are special because no tRNAs have anticodons for these sequences. Upon aligning with the A site, the stop codon is recognized by protein release factors that resemble tRNAs. The release factor instructs peptidyl transferase to add a water molecule to the carboxyl end of the P site amino acid. This reaction forces the P site amino acid to then detach from its tRNA, and the newly made protein, or polypeptide, is released. The small and large ribosomal subunits then dissociate from the mRNA and from each other. They are then almost immediately recruited into another translation initiation complex, ready to take on the next strand of mRNA that leaves the nucleus. After many ribosomes have completed translation, the mRNA is degraded so the nucleotides can be reused in another transcription reaction. So now we have our polypeptide. This means that our forming protein now has all of its amino acids, but its journey is not over yet. Recall back to Unit 0 material. The shape of a protein is very, very important to its function. So now there is a polypeptide in the cytosol or rough endoplasmic reticulum of the cell, and this new polypeptide must fold into a distinct 3D shape in order to function properly. Some polypeptides go on one step further and go on to join with other polypeptides to make a multi-part protein. Now, many proteins are actually decently good at folding on their own, depending on the environment that they're in, but some polypeptides need some help with proper folding. So, in the cell, we have helper molecules that assist in protein folding, and we call these molecules chaperones. Some proteins also contain special amino acid sequences that direct them to certain parts of the cell. These sequences, often found close to the ends of the polypeptide, can be thought of as the protein's quote-unquote train ticket to its final destination in the cell. We'll talk more on this process in our next discussion. But for now, tying this all back together with what we discussed earlier, we have our T cell. In the nucleus of that T cell, we had DNA. Within that DNA, we had a gene that had directions for making proteins, specifically cytokine proteins. And so we transcribed that one gene into mRNA so the mRNA could leave the nucleus where it was taken up by a ribosome. So the ribosome could then translate that code into a functional protein, specifically our cytokine proteins. And now there is assisted immune responses within this cell that can help out with the larger organism that the cell is contributing to. Make sense? And this process is also the same that would be followed when you're looking at why somebody's eyes are darker than somebody else's, right? The genes in the DNA code for proteins, and then those proteins can contribute to things like eye color. And so while in this example we used a protein that has a specific function, some functions of proteins are related directly to phenotype. I hope you found this helpful, and we'll talk more about how we transfer proteins all around the cell moving forward.